Okay, thank you for joining this webinar. I'm Takahiro Osada from Juntendo University, Tokyo, Japan. It is my pleasure to give a talk at this webinar. In my presentation, I will talk about transcranial ultrasound stimulation, TUS, a new non-invasive brain stimulation technique. This technique is capable of stimulating both cortical surface and deep brain structures. So my presentation consists of 4 parts. In the first part, I will give a brief introduction of TUS. In the second part, I will explain the feasibility of TUS by giving an example of MEP changes induced by TUS to the primary motor cortex M1. In the third part, I explain the TUS results of subcortical and cortical stimulation for response inhibition, one of executive function. In the last part, I will address my perspectives on future direction of TUS. The first part. So before going to TUS, I would like to address conventional non-invasive brain stimulation, NIBS method. There are several methods for conventional NIBS. For example, transcranial magnetic stimulation, TMS, uses magnetic fields for stimulation. Maybe many of you are using TMS. Other methods use electric fields for stimulation, such as transcranial direct current stimulation, TDCS, and transcranial alternating current stimulation, TSCS. These are collectively referred to as transcranial electrical stimulation, TES. TMS is widely used as a non-invasive stimulation technique. In this method, a coil is placed on the scalp, and the magnetic field generated induces an electric current in the brain. This is a powerful tool for intervention. However, it has a limitation. Its effects are primarily confined to the brain surface, as the magnetic field cannot easily reach deep brain regions. Similar challenges in stimulating deep brain structures are also encountered with TES. This limitation of conventional NIBS can be addressed by using ultrasound stimulation. In particular, focus ultrasound enables selective stimulation of deep brain regions. This is achieved through acoustic waves generated by piezoelectric transducers. The ultrasound waves can be focused precisely at the deep brain location. This is used for non-invasive brain stimulation. This will be the main topic of this webinar. In this talk, I will deal with low-intensity focus ultrasound, LIFU. In this method, ultrasound waves with low energy are emitted. LIFU is used for reversible neuromodulation. LIFU is distinguishable from high-intensity focus ultrasound, HIFU. In HIFU, heat caused by high-intensity ultrasound is used for therapeutic process, such as ablation. HIFU is basically an irreversible method, not reversible. For example, HIFU is used for treatment of tremor by applying to the thalamus. This is routinely administered within MRI scanner, MRI-guided focus ultrasound, MRG-FAST. After the treatment by administration of ultrasound to the thalamus, symptoms of tremor are reduced. Now, from now on, I'd like to focus on low-intensity focus ultrasound, LIFU. There are many terms to describe ultrasound neuromodulation: transcranial ultrasound stimulation, TUS, focus ultrasound stimulation, FUS, transcranial focus ultrasound stimulation, TFUS, and low-intensity pulse ultrasound stimulation, LIPUS or LIPUS. These terms indicate the same technique. As far as I know, there is no consensus about the terminology. But personally, I prefer the term TUS. This can be contrasted with TMS, a transcranial magnetic stimulation. I will use this term TUS in this presentation. There is a photograph. This is a photograph of TUS system, NeuroFast from Brainbox, equipped in our lab. Ultrasound waves are emitted from a transducer. Alternating electricity is generated from a drive system called a transducer power output, TPO. A typical transducer is a curve in shape. Ultrasound waves from the transducer are focused in space like this. A typical sonication focus is an ellipsoid. In a multi-element transducer, ultrasound waves were generated from each element. By changing the timing and phase from each element, the position of the focus can be controlled. In our system, we use 4 annular array transducers. The focus length ranges from approximately 30 millimeter to 70 millimeter. The size of the focus depends on the size of the transducer and the fundamental frequency of ultrasound. In a system of 500 kilohertz, the size of the focus is approximately 3 millimeter in lateral direction, the short direction, and approximately 10 millimeter in depth direction. As commonly employed in other ultrasound techniques, the ultrasound waves are generated in pulse manner, like this. This is a picture during our experiment. A transducer is placed on the scalp of the subject. As in the diagnostic ultrasound, hydrogel is placed on the scalp and inside the hair of the subject. It is because ultrasound waves cannot propagate in the air. In some cases, a hydrogel pad is placed between the transducer and the scalp. Because of the high spatial precision of the system, neuronavigation system is required to target the desired structure in each subject's brain. These are tracking markers for transducer and tracking marker for the head of the subject. For precise targeting, it is important to combine various methods to conduct TUE experiments in humans, like TUS, MRI, neuronavigation, and TMS, and also simulation. So why are neurons functionally modulated by ultrasound? A precise mechanism is not yet fully understood. However, the effect of ultrasound on the cell membrane is suggested to be a key for neuromodulation. Several mechanisms are proposed. One is acoustic cavitation, and another is thermal effect. However, the contribution of these effects are considered to be minor. The most likely mechanism is acoustic mechanical force. This mechanical force affects mechanosensitive receptors and perturbs the cell membrane. The change in ion channel permeability occurs. These changes alter neuronal excitability. Basically, TUS with low-intensity focus ultrasound is safe. But two safety concerns must be addressed. One is cavitation, or bubble formed by positive and negative pressure. These are termed non-thermal effect. The other is thermal effect. The FDA has established a guideline for ultrasound safety, but this was designed for diagnostic ultrasound rather than TUS. The values are not directly applicable to TUS. Too high for ISPPA for the intensity, for the non-thermal effect, and ISPTA too low for the ISPTA for the thermal effect. Scattering and absorption of ultrasound waves occur in the skull. It is important to estimate the intensity inside the brain and the thermal rise in the brain and skull. This can be achieved through acoustic simulation. When the ultrasound wave is applied to the brain, a focus comes here. The intensity in the brain decreases compared to the water condition because of the existence of the skull. Also, numerical simulation can be performed for thermal rise. After the ultrasound waves are applied for 40 seconds, the temperature rise is approximately 1 degree in the skull and 0.6 degree in the brain. To establish the guidelines, an international consortium called ITRUST, International Transcranial Ultrasound Stimulation Safety and Standards, was formed by TUS researchers from around the world under the strong leadership by Dr. Leonard Verhagen from Dantes Institute. I am a member of ITRUST. ITRUST consists of multiple working groups, including safety, reporting, practice, and so on. Safety recommendations have been developed by the Safety Working Group. This was published in the preprint server archive, and this recommendation is now set to be published in an academic journal. Also, the reporting working group's consensus on reporting standards was published in Brain Stimulation. The safety recommendation addressed two key aspects for mechanical effects. The mechanical index, MI, should be lower than 1.9. For thermal effects, the limits are set for the thermal index for cranial, TIC, or the temperature rise should remain below 2 degrees. The practice working group developed a practical guide under the tremendous leadership and efforts by Keith Murphy Stanford and Taylor Bergman Mainz. I am a member of this working group. This guide was recently accepted for publication in Clinical Neurophysiology. And you can also find a print version at archive. So now go on to the second part, feasibility. So now I'll explain the application of TUS for neuromodulation. This is a seminal paper by Win Ligon and Jamie Tyler at Virginia Tech, published in Nature Neuroscience in 2014. Ultrasound was applied to the human somatosensory cortex, and this causes SEP changes. This is also an interesting study by Win Ligon. TUS and TMS were applied simultaneously, the concurrent stimulation. MEP caused by TMS was evaluated between TUS condition and sham condition. MEPs were suppressed during sonication by ultrasound. This study by Leonard Verhagen, he was at the respiratory lab at Oxford. And so in this report, this study reported a long-lasting protocol for TUS with the sonication of 40 seconds. TUS was applied to the macaque brains, and the effect lasted approximately 2 hours. They observed resting state functional connectivity changes. Connectivity profile changes were specific to the stimulated sites. So we adopted this long-lasting effect by using this protocol. So to test the feasibility of TUS in humans, we examined the cortical activity changes after TUS by measuring motor evoked potential, MEP, with TMS to the human primary motor cortex, M1. Okay. The target location is crucial for TUS, so we identified FDR representation in the M1 via task fMRI. Subjects were instructed to perform a finger movement task inside the MRI scanner. They moved their left or right FDI muscle at 2 Hz as the arrow blinked for 20 seconds. Left, right, rest cycle was repeated 6 times. fMRI analysis revealed activation for each subject. The contrast of moving right FDI minus moving left FDI revealed activation confined to the region anterior to the central sulcus in the left hemisphere. There was variability in the depth of the FDI-M1 relative to the cortical surface. In some subjects, the FDI-M1 was located at the cortical surface like this. In others, the FDI-M1 was located deeply in the central sulcus. This is the distribution of distances, the distance between the cortical surface and FDI-M1. and the distance between scalp and the cortical surface, and the distance between scalp and FDAM1. In most subjects, FDAM1 was located near to the cortical surface. The median was 2.7 millimeters, and 80% of the population was located within 6 millimeters from the cortical surface. In this way, activation site for FDAM1 was identified for each subject. Next, the identified FDAM1 spot in the left hemisphere for each subject was targeted by TMS and TUS with the aid of an online navigator. MEPs from the right FDA muscle were measured before and after TUS using a single pulse TMS. A 40-second train of pulsed 500 kHz ultrasound consisting of 30 millisecond bursts every 100 milliseconds was delivered to the target FDAM spot. Intensity simulation revealed that the target site was effectively stimulated, as shown in the compared to the task activation. This is the MEP profile before and after TUS. MEPs were suppressed sustainably for at least 16 minutes. For the limitation of the time, all the subjects did not experience the experiment afterwards, but in some subjects, the MEPs recovered after the 90 minutes after the application of TUS. These results demonstrate that TUS induced the sustained suppression of cortical activity. Sustained suppression by TUS. Okay, so the third part, the response inhibition. Response inhibition supports adaptive behavior by suppressing inappropriate behavior. In the real-world situation, imagine you were crossing the street. When you notice a car coming, you need to rapidly stop actions, even after the initiation of actions. You stop stepping into the street. That is response inhibition. Classically, based on neuropsychological evidence such as studies by Adam Aron and Trevor Robbins, the right inferior frontal cortex, IFC, is responsible for response inhibition. Damage to the pars opercularis and pars triangularis causes deficit in response inhibition. Furthermore, response inhibition is known to be supported by the hyperdirect pathway connecting the prefrontal and subthalamic nucleus. Via a hyperdirect pathway. The stratum in the indirect pathway is suggested to be involved in response inhibition. However, how the prefrontal striatal indirect pathway contributes to response inhibition is poorly understood. Of course, TMS is a powerful interventional technique, but TMS cannot stimulate the deep brain structures such as stratum or STM. Instead, TUS can overcome this difficulty. To investigate response inhibition, subjects performed a stop signal task. The task comprised of go and stop trials. In go trials, a left or a right pointing arrow was presented as go signal, and the subjects were instructed to press a button indicating the corresponding direction. In stop trials, a go signal was followed by a stop signal after a stop signal delay, SSD, and the subjects were instructed to withhold their manual response. I mean, not to press a button. The SSD was updated with each stop trial based on performance, allowing to maintain accuracy of stop trials at approximately 50%. Task performance was evaluated by a behavioral index called stop signal reaction time, but not accuracy of stopping. SSRT approximately equals to reaction time of go minus SSD. And this SSRT is estimated based on an integration method. Shorter SSRT means more efficient in stopping. In contrast, longer SSRT means less efficient or poor stopping performance. This is the fMRI activation during response inhibition for the activation of the cortical surface and subcortical regions. In the subcortical regions, activation was observed in the STN subthalamic nucleus and the anterior part of the epithelium in the striatum. Next, activated site in the basal ganglia, the STN and the anterior epithelium, was targeted by TUS. The experimental schedule was similar to that in MEPs. 40-second TUS was applied to the target, and task performance was examined before and after TUS. We targeted STN and anterior epithelium in the right hemisphere. These were activated in fMRI. As a control, posterior part of the epithelium was also targeted. This site had no activation during the task. This is the task performance and the SSRT before and after TUS. Sustained SSRT increase was observed after TUS to the STN. This means disruption of response inhibition performance. Also, TUS to the anterior epithelium induced sustained SSRT increase. In contrast, TUS to the posterior epithelium did not show significant disruption of performance. Next, we explored the anatomical connections between the basal ganglia and cerebral cortical regions. We employed the diffusion MRI data. When the seed was set to the anterior epithelium, the tracts were estimated using probabilistic tractography. This shows the connected areas with anterior epithelium. Structural connectivity data was then thresholded with task activation via fMRI. The anterior IFC, AIFC, in the right hemisphere was identified as the area that had robust structural connectivity with anterior epithelium and task activation. Next, we examined the essentiality of AIFC, the identified area. Middle frontal cortex was also examined as a control, MFC. MFC showed task activation, but did not show structural connectivity with the anterior epithelium. TUS to the AIFC elicited the sustained SSRT increase before and after TUS. In contrast, MFC did not show disruption of task performance. The AIFC is located at the anterior edge of the pars triangularis. The pars triangularis is located anteriorly to the pars opercularis. Most of the previous studies in response inhibition highlighted the pars opercularis, including the ventral posterior IFC, VPIFC. The AIFC was not highlighted in the previous literature. So interim summary for part 3. We applied sustained suppression effect with TUS. The anterior epithelium showed the causal contribution to stopping. The AIFC is the counterpart of the anterior epithelium, and the AIFC anterior epithelium circuit works as an essential route for stopping in the indirect pathway. So now I would like to further discuss the neural circuits for response inhibition. The IFC inferior frontal cortex, particularly the ventral posterior IFC, VPIFC, plays a critical role in response inhibition. The IFC interacts with the basal ganglia and subsequently the primary motor cortex. Perceptual information, particularly visual input, serves as a trigger for response inhibition. However, there is no direct pathway connecting the visual cortex and IFC. This indicates that the existence of an intermediary area between visual cortex and IFC. To identify this intermediary area, fMRI and the diffusion MRI were utilized for pathway tracking. To identify the input areas to the ISC, diffusion MRI was used to track regions connected with BPFC. Structural connectivity data was then thresholded based on the task activation. 19 areas were identified as having both structural connectivity with the BPFC and task-related activation. Among these 19 areas, the dorsal anterior insula was the only region connecting with the V1, primary visual cortex. So next, we tested the essentiality of the identified DA in dorsal anterior insula. As the DA is located deep within the lateral sulcus, TUS is suitable technique for targeting this region. The DA was stimulated for certain suppression with a 40-second stimulation by ultrasound, and the performance of the self-signal task was monitored before and after TUS. As a control, ventral anterior insula, VA-ins, was also stimulated. This region was activated during the task, but this was not connected with the BPFC. After TUS to the DA, an increase in SSRT was observed, indicating impaired stopping performance. In contrast, TUS to the BA-ins did not disrupt the performance. We then explored the regions connected with DA-ins and basal ganglia. 3 criteria were set: structural connectivity with the DA-ins, task activation, and structural connectivity with any of the basal ganglia, SDN, anterophthalmian, and caudate nucleus. These basal ganglia regions are observed to be activated during response inhibition. As a result, 2 regions, the BPFC and AFC, were found to meet these criteria. The BPFC and the AFC was postulated to be located at the downstream of the DA-ins. To investigate the insula-IFC interaction, we combined TUS with single pulse TMS. If the DA-ins is at an upstream position, sustained suppression of the DA-ins by ultrasound would negatively affect the BPFC and AFC. This would reduce the transient disruption effect by single pulse TMS. TUS was administered to the DA-ins for sustained suppression. Before TUS to the DA-ins, single pulse TMS to the BPFC and AFC caused a disruption effect, transient disruption effect, as indicated by a prolongation of SSRT. The SSRT was compared between the condition of single pulse stimulation and no stimulation condition. So for TUS, so we observed both the prolongation in single pulse TMS to the BPFC and the AFC. However, after TUS to the DA-ins, single pulse TMS to the BPFC and AFC no longer induced the significant prolongation of SSRT. These results confirmed the causal influence of the DA-ins on both the BPFC and AFC. Finally, we investigated the relationship between the downstream areas, BPFC and AFC. Same protocols of TUS and single pulse TMS were applied to these regions. TUS was used for sustained suppression, and the transient disruption effects were compared before and after TUS by single pulse TMS. Before TUS, single pulse TMS to the BPIFC and AFC resulting in a significant prolongation of SSRT. After TUS to the AFC, single pulse to the BPIFC still caused a significant prolongation of SSRT. In contrast, after TUS to the BPIFC, single pulse TMS to the AFC did not result in a significant prolongation of SSRT. These findings suggest that the existence of a unidirectional causal influence from the BPIFC to AFC and no causal influence from the AFC to BPIFC. Summary of this part 3. Perceptual information is initially processed in the primary visual cortex before being relayed to the dorsal anterior insula. From dorsal anterior insula, the information is transmitted to either ventral posterior IFC or anterior IFC, and subsequently to the basal ganglia and primary motor cortex. Within the IFC, a unidirectional influence from the BPIFC to the AFC was identified. Okay, so let's go and move on to the final part. I'd like to address the future directions of TUS. So first, I'd like to discuss the effect duration of TUS. As demonstrated in the data on MEPs and task performance, TUS with sonication duration of 40 seconds induced sustained suppression of activity lasting at least 60 minutes. For clinical applications, achieving a much longer effect duration with TUS is desirable. This requires a deeper understanding of the biophysical and physiological mechanisms underlying neuromodulation, as well as optimization of stimulation protocols and parameters. As I explained, the detailed mechanisms of ultrasound neuromodulation are not yet fully understood. At the cellular level, research has reported the role of mechanosensitive channels, such as TRP channel, in neural membranes. Another study highlighted the involvement of astrocytes for neuromodulation by ultrasound. Additionally, research showed cell type-specific activity changes induced by ultrasound. Further research is required to clarify these biomechanisms in greater detail. Next, I'd like to discuss the directionality of TUS effects. In this talk, I primarily focused on the suppression effect of TUS. Recently, several labs reported alternative stimulation protocols for achieving offline effects, particularly enhancement effect. One such protocol is theta burst stimulation, developed by Robertson Lab at Toronto. In this protocol, pulses were delivered at 5 Hz, corresponding to the rhythm of theta, for a duration of 80 seconds. When this TBTUS, theta burst TUS, was applied to the primary motor cortex, an enhancement in MEPs was observed. The effect of TBTUS were supported by MRI spectroscopy in a study by Transcranial Lab at Plymouth, UK. Theta burst TUS was applied to the posterior cingulate, PCC. Reduction of GABA concentration was observed after TUS application, TBTUS application. This decrease in inhibitory neurotransmitter is associated with an enhancement of activity. However, there is currently no theoretical or biophysical explanation for this phenomenon. Further replication of these experiments is required. And if this protocol can be used for regions beyond the primary motor cortex, TUS could potentially be utilized for bidirectional manner, both for suppression and enhancement of activity. So to utilize, fully utilize the power of TUS, we have to require the deeper understanding of biophysical and physiological mechanism of ultrasound neuromodulation. And of course, it is required to optimize the stimulation protocols and stimulation parameters. So, as I mentioned, it is preferable to determine the target location based on fMRI activation. Connectivity data provides additional support for target planning. The high focality of TUS further enables a personalized high-precision intervention. This concept holds the potential to advance non-invasive clinical treatment using TUS. So in this talk, I presented the conventional approaches of MRI and TUS. MRI serves as an imaging technique for observation and measurement. On the other hand, TUS functions as an intervention technique. By leveraging the unique strengths of each method, I believe we can identify the neural circuits underlying specific cognitive processes and elucidate the principles governing their operation. This approach holds the potential to pave the way for neural circuit-based diagnosis and treatment for neuropsychiatric disorders. Okay? These are collaborators, and most of the work was performed under the supervision of Professor Seiki Konishi, and basal ganglia stimulation was performed mainly by our graduate student, ex-graduate student, Dr. Koji Nakajima. These are collaborators, and these are grants supporting our research. Okay, thank you for your kind attention.